My name is Kunthearithy Heng, the author of this research, and my advisor is Associate Professor Chitlada. We are both from Thammasat University, and my another co-author is Associate Professor Punkit. He is from Ramathapadi Hospital. Today, I am very happy to welcome you all to my research presentation. My research title is a histological study on the effect of 1550 nanometer erbium class fractional laser treatment of androgenic alopecia. This is my presentation outline, and I will present accordingly. AGA is one of the non-scarring hair loss disorder, which caused by multiple etiology, including genetic, hormone androgen, and microinflammation of the perifollicular area. AGA has a major impact on individual self-esteem through psychological health. It is characterized by the progressively shortening anagen and prolonged telogen and miniaturization of the scalp hair follicle. Male generally present with recession of hairline at the temple and vertex balding, while female is typically diffuse, seen over the crown area with preserving the frontal hairline. In Thailand, the prevalence of male pattern hair loss is about 38.52%, while the prevalence of total population around the world is increased with age. The standard treatment of AGA is a topical solution minoxidil and a systemic 5-alpha reductase inhibitor, which is still limited by its low efficacy and undesirable side effects. Extensive study has recently been conducted to search for an effective AGA treatment strategy, including utilizing a fractional laser. A 1550 nanometer erbium class fractional laser is one of the new effective treatment options, showing a maximum therapeutic effect and fewer side effects. The mechanism of this laser work on the hair growth is thought to be the part of the wound healing process. Hair growth may follow the induction of some level of wound healing, which means there is something behind we should know about a wound-induced hair growth. So what about macrophage? What is related? If we talk about wounding, even from laser or injury, we might notice on many studies that has been conducted on inflammatory cells at the site of injury and wounding. During wounding, monocytes are recruited and attracted to the wounded tissue, where they differentiated into macrophages. Macrophages in the wounded area contribute to the an acute inflammatory response, and also contribute to at the late state of wounding, and produce various growth factors such as insulin-like growth factor, fibroblast growth factor, epidermal growth factor, and hepatocyte growth factor. Those factors play role in the influencing hair growth and the hair cycle in the wounding area. According to the recent study, Osaka and Casalina also reported that macrophages play an essential role in wounding-induced hair growth and identify the link between macrophages and hair follicle cycling. On the other hand, if we talk about fractional laser, Lee et al. And Kim et al. In 2011 reported a significant increase in hair growth and hair density after 1550 nanometer erbium class fractional laser. Hair growth followed the induction of some level of wound healing and after laser irradiation. However, in marine study, reported that anagen entry usually occur after laser treatment at a pulse energy of at least 10 millijoule with density of 1,550 microthermal treatment zone per square centimeter, and the density of the 1,600 microthermal treatment zone per square centimeter normally increase inflammatory cytokine and expression. Move to the objective. This study aimed to evaluate the mechanism of 1550 nanometer erbium class fractional laser by comparing the difference in histology of the scalp biopsy before and after treatment. Let's move to the methodology. This study was designed as a retrospective pilot study with a total of 12 samples. The inclusion criteria is the specimen from a subject that show an improvement from AGA treatment with 1550 nanometer erbium class fractional laser. The exclusion criteria are listed below as shown in this slide. Since this is the retrospective study, I would like to mention briefly about the previous one. The previous study was conducted in 2017 and enrolled 23 male and female AGA patients using 1550 nanometer erbium class fractional laser with following parameter of 2 × 12 mm tip, 6 millijoule power energy, 300 watt per square centimeter density, and static mode 2 to 4 passes at the balding area for without local anesthesia for total of 14 sessions in a 2-week interval for 24 weeks. Scalp punch biopsy was performed at the baseline 1, 2, and 3rd month after the first laser. Each group of months contained 4 patients. Those histological samples were cut transversely into 3 micron paraffin embedded and was stored for examination. In this study, we performed immunohistochemical analysis to evaluate macrophages. We sent the tissue to the company, so the specimen were diparaffin and stained with CD68 for which is the marker for macrophage. Those specimens were then evaluated using an evaluator score, which it was directly compared before and after treatment slide. It then were scored as negative or positive. If there is a negative, it would have a 0 score. Or if there is a positive, the intensity of the staining will be was graded on the scale of 1+, 2+, and 3+. The control of the positive and negative are look like this. This slide overall show an example of the company that follow during the experiment. About the result, these the 3 groups, including baseline 1st, baseline 2nd, and 3rd month, did not show remarkable alteration. All over tissue had a 0 score. This slide shows that there is no infiltration of macrophage before and after laser irradiation. So according to the literature review above, the possible mechanism of laser wound healing hair growth included increased blood flow, induction of inflammatory cytokine and growth factor, direct stem cell, bud cell, and dermopapilar cell stimulation, produce various molecules that play a role including member of fibroblast growth factor. Those are recognized as the main factor that influence hair growth and the hair cycle. Importantly, several studies has already discussed about macrophage that infiltrate upon wounding, and macrophage are needed for hair follicle growth. Since the macrophage contribute to the cyclic activation of hair follicle stem cell. There is also a connection between macrophage and regulation of the hair follicle cycling. And macrophage play a fundamental role during hair follicle cycling. Those were mentioned above improve our understanding of the relationship between macrophages and hair growth, which cannot deny. However, we failed to determine macrophage alteration. We consider that our study has a limitation during experimental. The laser parameter used in our study might not be sufficient to stimulate inflammatory cell infiltration into the skin. The molecular change in the wounding response caused by thermal injury might be depend upon laser energy. The histological section from the scalp tissue might not be the optimal depth, as the previous study showed the macrophage located near the distal of the hair follicle. For conclusion, a total of 12 samples of AGA patients with an improvement from laser treatment were enrolled in this study. Immunohistochemical analysis were performed using CD68 to evaluate macrophage. There was some mistake we might have made during our experiment. The laser parameter might not be sufficient to stimulate inflammatory cell infiltrate, and the histological specimen will not be cut at the optimal depth to identify macrophage. Based on our knowledge, to dismiss this limitation, we would suggest for the study using optimal laser parameter and the proper histological section should be conducted. Thank you for your attention.
Any participant would like to share or have some comment?
So I'm not clear about your research study. Yes. Since
you said that
in your study you don't find any macrophage in the lesion, and you conclude that it is the error from your technique. Why don't you think that the real thing?
It is a real thing.
It is a real thing.
Sorry, Ajahn.
Yeah. Let me repeat
my question again.
Yeah.
Because you said
that all of your
I think it's very
echo occur in
why I
talking. Yes.
It is because Ajahn Chutima and Ajahn Vorachai is in the same room, and when Ajahn Vorachai talk, it's going into the microphone of Ajahn Chutima.
Oh, I see.
Yeah.
So what can we do?
So you need to close one of the microphone.
Yes.
And use just only one microphone. Yes.
Close your microphone. If close my microphone, I cannot speak anymore.
Yeah.
Okay.
Already closed, but still echo.
Let me say again. It's better.
It's better right now. Yeah.
Yes. As you said that your research you did not find a macrophage in the lesion, right?
Okay,
that's because of your maybe techniques or the tissue not represent the areas. Why don't you think that this is the real thing that occurred? And in my opinion, if you found macrophage in the lesion, it doesn't mean anything, because in the wound healing, you will find macrophage there, because it is part of the chronic inflammation or the reparative process. And for the growing of the hair, if you find macrophage, it doesn't mean anything, except that you find that there is some specific cytokine that is different from what you find in the chronic inflammation, and that means something.
Yes, that is the reason why we thought that the techniques of our experiment might has a problem, because normally macrophages are dermal macrophages are represented in close proximity to the hair follicle, and also also contribute to the hair growth, and also the laser induced wound healing. We suspected that the macrophage will be increased in our specimen.
Yeah.
But our hypothesis go wrong, because macrophages did not up regulation. This is why we have to think that what is the main problem that caused this negative result. But it is good. Good.
I haven't seen. I'm pathologist. I haven't seen your section. But if I saw your section, I can suggest you something. I don't think that this is the real fact. If you find something, you have to explain in that way why it is. Macrophage may not be involved in this process. Is it possible? Because what you find, you have to believe that.
Yeah, Jan. Because we cannot find even from the before and after treatment.
Okay, thank you.
Thank you. Thank you, Professor.